In the last video, we had discussed on the development, on the formation of the antheridium. Okay, of the antheridium, that is the male gametes in both the macrandrous as well as the nannandrous species, where we see that in case of nannandrous species, there is a development of the dwarf male. Okay, and the antheridia, as you can see, they form on the dwarf male. So today we are going to discuss on the development of the female gamete. Okay, we will study on the development of the oogonium, that is the female reproductive structure. Now, the development of the oogonium in both the macrandrous as well as the nannandrous species, they are similar. Okay, in monoecious species, monoecious meaning where the both the antheridia and the oogonia, they are on the same filament. So in monoecious species, the antheridia and oogonia, they are present on the same filament. But in dioecious species, we know that they are borne on different filament. In one filament, we will find the oogonium, and in the other filament, the antheridium. Now, development of oogonium, like I had mentioned just now, that is, however, similar in both the macrandrous as well as the nannandrous species. And here, so let us study about the development of the oogonium. Any cell, okay, any cell in the filament, any vegetative cell which has the apical cap, okay, which has the apical cap, like in this case, you can see this cell, it has two apical caps here. So any cell with the apical cap, they may function as the oogonial mother cell. So this acts as the oogonial mother cell in sexual reproduction. Now this cell divides transversely into the upper oogonium and a lower, as you can see here, this whole cell filament, which has a cap cell, it divides into two cells, you can see here. The upper cell, which acts as the oogonial mother cell, and the lower cell, it acts as a suffulatory, or we call a supporting cell. Okay, now this supporting cell, it may again function like the oogonial mother cell, and it will divide again. Okay, this supporting cell, it can divide again to form into two cells, giving rise to one oogonial mother cell and another supporting cell. So it keeps on dividing. And in such cases where this supporting cell, it divides 2 to 3 times, we get 2 or more oogonia are formed. Okay, that means 2 or more oogonia are present in a single filament. Now, initially, the oogonium, it is wider. Okay, as you can see here, it is wider when you compare it with other vegetative cells, or here if you compare it with this supporting cell also, the oogonial mother cell is slightly wider. But at maturity, it swells. As you can see in figure B here, at maturity, figure B and C, it swells and it becomes globose. Okay, and its protoplast, it forms, here in figure C, you can see the protoplast, it forms a single uninucleate non-motile egg. Okay, the egg is non-motile. It cannot move. It remains in a single place. Now the young egg is usually green in color due to the presence of the chloroplast. But then at maturity, you can see here in figure C, okay, at maturity, what happened to the oogonial mother cell? There is a development of this uninucleate non-motile egg. and the young egg is green in color due to the presence of the chloroplast, and also you can see a small pore or a transverse slit, as in this case this is the transverse slit, which develops on the oogonial wall. You can see it develops on the wall of the oogonial cell. Okay, now just opposite to this slit, just opposite to it, you can see here a hyaline area is present. A hyaline area is present in the egg, and this hyaline area is known as the receptive spot. Okay, it is known as the receptive spot because here when the antheridia or the male gamete it enters, it will enter through this slit. Okay, so this portion here, therefore it is referred to as the receptive spot, and the egg nucleus, it will move under the receptive spot. You can see here it will start moving just under this receptive spot before fertilization. And in some species, prior to fertilization, in some species you can see here a mucilaginous or a gelatinous material is extruded through the pore or through this receptive spot in order to facilitate the entry of the antherozoid into the oogonium. Okay, when the antherozoid or the male gamete, it will enter through this receptive spot. And what happened? The egg nucleus, just before fertilization, it will move just under this hyaline area or this receptive spot, and there will be a production of a gelatinous substance, a mucilaginous substance, okay, to facilitate the entry of the male gametes or the antherozoids into the oogonium. So this is the development of the oogonium in both the macrandrous as well as the nannandrous species.